You find your quiz? It's up. You don't have to do it until next Sunday. It's on Top Hat. Just open up Top Hat. It will be there right now. And it's open note and open book, right? So you should get 100. You might not. Huh?
Is it timed?
No.
Okay.
I'm also going to put another quiz up for you once I know how far we get in the brain. But I didn't want to put it up there yet because I didn't want you to cry. You'll be like, I don't know any of this. No, you don't yet, but you'll be fine. So go ahead. I have no idea what chapter numbers are ever. It's nervous system 1 plus a little bit of the stuff we're doing today. Okay? And then the next one will be over the stuff we do today and Wednesday. Okay? And it's just giving you guys a little practice. If you do not do it by Sunday, you will get a 0, and I am not taking it late because there's no point in doing it after you already had the damn test over the stuff that I was trying to get you studied. Right? Okay? So, everybody understand? Did you find it? Did you find it? Yeah, it's not hard to find. It's just right there in red, right? And it tells you that there are 11. It's really cool. But apparently on Top Hat, there's a way that, like, if you answer it wrong, I can redirect your answer and give you a hint. But I can't make it show up, so I have to email Top Hat. So maybe I'll figure that out by the time I post the next one, right? Because I wrote all of the little redirectors or hints, but then I tried to take it, and I got it wrong on purpose, and it didn't show up, so I don't know how to do that. Okay, so what else do I need to tell you? Oh, who has their practical today? They're going to do awesome, aren't they? All of them? Yes? Okay. You're going to do great. Your mom told me. Okay. So we're going to finish up this set of notes and start on brain today. Brain is going to be hard for you. Okay? It's hard. How do you make sure that you feel a little bit more confident? What would you do before Wednesday? Yeah, you would preview the material, and if you really wanted to be confident, you'd probably look at the video from last semester so that you didn't feel rushed and overwhelmed, right? That's probably what I would do. I would look at the video from last semester and see if I could master some of the stuff before I even get into class, okay? The terminology is unfamiliar, and there's like structure-function relationships, and the brain's kind of hard to picture 3D. I'll bring a couple models in for you guys on Wednesday when we actually get into brain structure, and you can look at stuff and hold them while we're going through. In addition, you haven't done it in lab yet, but really, so we won't finish the entire next set of notes. Wherever we finish on Wednesday, that's where I'm cutting off the material for the second test. Okay, so it probably will not get through all of nervous system number 2, just however far we get. Okay, all right. So. Last part on nervous system 1 is just regeneration of nervous tissue. So let's talk about what happens if you damage your nervous system. Do you ever get any regeneration? And the answer is, it depends on whether you damage cell bodies or axons. And then it depends on whether it's CNS or penis, right? So pretty much, if you damage neuron cell bodies, you're kind of screwed. You're not really getting any more of them back. Now, there are a couple of exceptions. We did find a couple of places in the brain where the neurons still have the capacity to go through mitosis, but pretty much just depend on the fact that you're not getting any of those neuron cell bodies back. Matter of fact, most neurons don't have centrioles any longer, so centrioles are necessary for cell division and nuclear division, right? Okay, so what about if you damage an axon? It depends on whether you damage an axon in the CNS or the PNS, okay? So let's talk about PNS damaged axons and what could happen. Okay, so we're looking right here, and this is the peripheral nervous system, and I can tell for 2 reasons. How can I tell for 2 reasons that this is the peripheral nervous system? Go ahead. Exactly. The myelin sheath is made by what kind of cells in PNS, remember? Schwann cells or neurolemocytes. And then remember, it's also going to a muscle, and you don't have any muscle in your brain last time I checked. Okay? Okay, so that's going to be PNS. Does that make sense? Okay, so this is PNS. Now, this is only showing you one axon. If I were in the PNS, that would be part of a nerve, right? It would be all packaged and bundled with thousands and thousands of its buddies. But we're going to simplify this. And we're going to say, okay, if you damage a single axon in the peripheral nervous system, first off, they're all myelinated in the peripheral nervous system, all of them. So if you damage it in the P and S, then what's going to happen is, first thing is the region distal to the injury is just going to die because it's like cutting an arm off of a body. It doesn't have access to metabolism and protein synthesis and ATP. It doesn't have access to any of that stuff any longer. Does that make sense? So that region is going to die. So this region will die. Let's say this is skeletal muscle. It looks kind of the way they drew it like smooth muscle, but just let's say it's skeletal muscle. When you lose innervation of skeletal muscle, what happens? You are paralyzed, right? And then eventually that damage can become permanent if you don't re-innervate. So as long as I haven't re-innervated, all of those muscle fibers that were stimulated by this neuron are paralyzed unless and until I get regeneration. So what's going to happen in the peripheral nervous system is the P and S will try to regenerate the tissue. First, the axon will start to grow from the site of damage, right? Okay, and then the neurolemocytes or Schwann cells, they'll make a growth tube for you. So they'll basically start to go through cell division and make a little tube for you to aim at, right? Trying to go into the tube. And we used to think it was just like physically aiming at it, and then we figured out that the tube actually secretes chemicals that actually attract the growing axons called chemotaxis. They're going to actually secrete some chemicals that make it more likely that you will go in through the tube. So it's going to basically form a growth tube. And then this doesn't happen very quickly, but the axon will grow 1 to 5 millimeters per day, which sounds really fast until you realize that the axon may have to go all the way here or all the way here, right? So if it hits the tube and you give it time enough, it can eventually reconnect to the effector, the skeletal muscle, and then it will work again, right? But this is really slow in long axons. It's 1 to 5 millimeters per day. After you have P and S nerve damage, it can take up to 2 years to know whether you're ever going to have reinnervation. And then remember that this is only one axon. In a nerve, do you think that all of the axons always hit their tube? No, they don't. So you can end up with partial innervation, right? You can still have weaknesses even after 2 years. And sometimes they hit the wrong damn tube too, and that's really weird. Because you think you're going to be actually moving muscle over here and it's over on this side, or you've got sensation that is, you know that you're touching right here, but it feels like it's happening right here. Does that make sense? If they actually hit the wrong tube when the reinnervation goes on. So it will reconnect to the effector. But the important part to know about this is if you ever have nerve damage, you have to keep this tissue alive for those 2 years in which you can't actually use it, right? So you have to go to physical therapy. You have to stimulate it. You have to move it around. If you don't, what your body will do is say, this is really energetically expensive tissue. How about I just replace it with something cheaper like connective tissue? And then you'll start to have permanent muscle atrophy and loss of muscle tissue, which is why after, for instance, let's say someone has a spinal cord injury. Spinal cord is CNS, right? And they've got paralysis because of the spinal cord injury. But does it make sense to you that usually when you have C and S damage because of the spinal cord injury, you might also have P and S damage? Yeah? Just that region? So if I say, oh, well, the damage is permanent, I'm never going to get any reinnervation, so I'm not going to go to physical therapy. Well, what if in 2 years you figured out that some of the paralysis was due to P and S damage? And then you would let the tissue die. Then you actually get no reinnervation. So you have to do exactly the thing you don't want to do after a spinal cord injury, is that you have to actually go move even when you can't move yourself, right? Okay, so if you reconnect to the effector, you can get reinnervation. Now, what about CNS? In the CNS, you're screwed because if I damage white matter in the CNS, it's not myelinated with neurolemocytes, it's myelinated with what? Oligodendrocytes. And here's what oligodendrocytes do when white matter is damaged. You damage white matter, they go, I'm out. They just pull up their little fingers, and they don't try to help at all. They don't make any regeneration tubes. And then your body won't leave like a vacuum. It won't leave an absence of space. So if you damage something, what happens is astrocytes move in and just like sort of start forming scaffolding. But they actually end up physically being in the way of the axon. So it's the brain's equivalent of scar tissue. Does that make sense? So if you damage white matter in the brain, it's just as bad, well, not just as bad, but it's just as likely to be permanent as it is damaging gray matter in the brain. Does that make sense? Okay, so now let's talk about protecting your brain, since apparently that's pretty damn important. Okay. So the brain and cranial nerves is what we're going to do. We won't get through cranial nerves, and those are good to reinforce in lab anyway. Oh, how did I accidentally close that? Give me just a second. I'm going to reopen all those images for you. Why did I do that? I don't remember doing that. Just talk amongst yourselves for a second, because I got to reopen these images that I just closed. You guys are a couple minutes ahead of the other class anyway, so. Okay. That's crazy. Okay. We're almost there. Sorry. Choroid Plexus, there it is. Okay, so, oh, there it is. That's the picture I'm on. So what we are going to talk about right now, before we actually get into the structures of the brain and related directly to what we just talked about, which is your brain pretty much doesn't regenerate almost at all. So how do we make it very, very unlikely that we are going to damage it or get infections in it? Because most of us, maybe even all of us, have never had a brain infection. Like damn near everything else has been infected at one point. But why have you not had a brain infection? Because there's multiple layers of protection around your brain and your spinal cord. And the first one is just the physical barrier of bone, right? So we can see in this figure right here that the bone that is actually surrounding the cranium is pretty darn thick. Do you see that? Okay. In addition, of course, you also have bone around your spinal cord. So I'll show you the spinal cords as well, even though I accidentally closed everything because I'm a dingbat today. Okay, so let's look at the spinal cord as well. So you remember that the vertebra actually sits in a bony case, right? Okay, so we've got bone around both the brain and the spinal cord, but then deep to the bone, we have 3 layers of membranes, and then deep to that, we have fluid that is surrounding the brain. And then in addition to all of that, we also have what's called the blood-brain barrier, which actually physically limits what can get out of the bloodstream into the brain tissue. This must be pretty effective, right? 7 billion people on the planet and very few brain infections as a proportion of those 7 billion people. So what we're going to do today is we're going to look at the protections around the brain and the spinal cord. So the first line of protection is, of course, bone. And then the second line of protection are what we call the cranial meninges or the spinal meninges. They are also around the spinal cord. And you're actually seeing them right here. So if we go from bone right here, right? And then we look at the surface of the brain. The surface of the brain usually looks kind of shiny. And the reason it looks kind of shiny is because there is kind of like a serous membrane, but it's not really like a serous membrane. There is a membrane that's on the surface of it and stuck to the surface of it. It's called the Pia Mater. And then there's another one right here that's going to be a little bit more fibrous. And then there's going to be a really, really thick, opaque one, right? So right now what we're going to do is we're going to go through these meninges, which are these layers that are around the brain. So let me just draw you a bad brain picture and spinal cord and say, okay, here's the brain, here's my spinal cord. First thing is, your spinal cord is shorter than you think it is. Most people think the spinal cord goes all the way down, but it actually ends about L1 or L2. Ends right about there. You have a layer of 3 layers of membranes that surround and are continuous around the brain and the spinal cord. So let's go superficial right next to the brain to, deep right next to the brain to superficial. So the one that is stuck to the surface of the brain, it's actually stuck, but I can't draw it stuck and have you see it. So it is going to be the reason that the brain is shiny. And it is called the Pia Mater. And Pia means gentle, and what do you think mater means? Mother. It's the gentle mother. I'll show you the Pia Mater. Let me find a cool picture. Not that one. Oh, there it is. Where did my brain go? There it is. Okay, so this is a fresh brain. I'll show you guys an autopsy of a fresh brain on Wednesday, so you'll get to see the actual texture of the brain. But do you see how it's nice and shiny right here? That's the Pia Mater that's on the surface of the brain that you're actually seeing right there. The brain is really, like, the texture is so much different than the preserved brains that you get. The preserved brains that you've seen have been injected with straight formaldehyde because they're really fatty and they start to disintegrate almost immediately. But a fresh brain, if you hold it on your hand, it'll start to slide off the sides. It's basically kind of like a bag of pudding, really. It is so soft that when they do brain surgeries, they actually don't even use scalpels all the time. Sometimes they just stick in a little suction thing like at the dentist office and suck out a little chunk, right? So it's way softer than you imagine it to be. Okay, so that's the Pia Mater, and then we are going to move superficial to the Pia Mater for the next layer. The next layer is more fibrous, and this is called the arachnoid. What does that remind you of? Yeah, sometimes called the arachnoid mater, and sometimes they leave off the mater. You are seeing the arachnoid in this image right here, where it looks kind of fibrous, right? This was shiny, that was the Pia Mater that you're seeing, and this looks more fibery. That's the arachnoid. And then superficial to the arachnoid is going to be the densest, thickest one. It's generally opaque. If it's intact, you won't even be able to see the structures of the brain. So this guy right here, that's the most superficial one, is called the Dura Mater. And Dura means tough, so this is literally the tough mother. It's a tough mother. So this is the Dura Mater that you're seeing right here, right there. It's opaque. It is really, really thick, dense, irregular connective tissue. It's really hard to puncture on accident. Okay, so here you guys are going to do a sheep brain dissection next week. You're actually looking at the Dura Mater on a sheep brain because you can't see any of the brain structures, right? You have to take the dura off in order to see the internal brain structures. So as long as the dura is still on there, you're not going to really see anything. Okay, so let's get a little bit more detail about each layer as we go through. Okay, so we've got the cranial meninges. Singular is meninx. Superficial to the cranial meninges around the spinal cord is a space called the epidural space. So you guys have heard of epidural injections before, yes? Okay, so let me teach you what that has to do with this story. So if I draw a vertebra around here, yeah? Superficial to the Dura Mater is the epidural space. And around the spinal cord, the epidural space is filled with loose connective tissue and fat. Do you see? And so it provides a little padding for the spinal cord. That's not true around the brain. around the brain, the Dura Mater is actually continuous with the periosteum of the skull, so there's no epidural space up here. So, you've heard of epidural injections before. What you do with an epidural injection is that you inject an anesthetic into the epidural space, right? I'm not trying to touch the central nervous system, right? Otherwise, I would go through the dura, right? I want to stay epidura. So I inject it into the epidural space, and the anesthetic diffuses out through the fat tissue into the nerves that are coming out of that region. And if you do it down here, the nerves that are coming out of the lumbar region innervate the legs and the abdomen and the pelvic cavity, right? Does that make sense? It's an inexact science though, guys, because it's just based on diffusion. If you stick your butt on a pillow, it'll anesthetize you all the way up to here. You lay on your right side, it'll anesthetize the right side more than the other. Lay on the left side, it'll anesthetize the left side more than the other. Because really all it's doing is sort of diffusing through the epidural space into the nerves that are coming out in that region, right? Does that make sense? Okay, so that's what an epidural is. Now, the epidural space is only present in the vertebral cavity. So let's talk about the cranial cavity, what you are actually going to see in the cranial cavity. So in the cranial cavity, what you are going to see is the Dura Mater has 2 layers in the cranial cavity. It only has 1 elsewhere. The superficial layer of the Dura Mater in the cranial cavity is continuous with the periosteum of the bone. It's attached to the periosteum of the bone. So it's called the periosteal layer of the Dura Mater. And the deeper layer, what you're actually seeing right here, the deeper layer is going to be right here, and that's going to form that sac that's around the outside. So let's talk about the periosteal and the meningeal layer. Okay, so let's look at this figure. We're going to be looking at this figure quite a bit today, okay, and orient you to what you're looking at and find the periosteal and the meningeal layer. Okay, so if you look right here, you're seeing the skull, right? And you see this line that's immediately deep to the skull. That's the periosteal layer of the Dura Mater. And then you've got this blue stripe. That blue stripe is actually a blood vessel. There's blood vessels that run between the periosteal layer and the dural layer, the meningeal layer of the Dura Mater. You're going to learn the names of those blood vessels. I won't test you on them on this test because I want you to do them in lab first, okay? But those blood vessels are draining blood from the brain that has already been used, right? And so those blood vessels are going to be in the dura between the 2 layers. So they're called dural venous sinuses. So let's break that down. Dural because of where they are. Venous because they are veins, not arteries, yes? And a sinus is just a big vein, okay? So I'll show you these. Now realize I'm not going to test you on the names of them yet, but I want to show them to you. Here's one of them. This one's called the superior sagittal sinus for obvious reasons, right? It runs right under the sagittal suture. It's called the superior sagittal sinus. But there are more. I'll show you some other ones too. Let me find chapter 15. Okay. So let's find those. Here they are. Okay. Here are a bunch of them. See these big blood vessels? What these big blood vessels are doing, guys, is draining blood from the brain, and ultimately they are going to dump it into the jugular veins that go through the jugular foramen, and that's how blood gets out of the brain, right? That's how it goes back into circulation. And what these are doing is the blood is going that direction toward the back, right? Because you know where the jugular foramen are. They're at the bottom and the back of the skull. All of these are coming together, and they're going to dump into there. Does that make sense? Okay. So for right now, you don't have to know the names of those vessels. You will learn them when you do the brain. Okay. All right. So these are called the dural venous sinuses. Okay, and that's right there. The only one that you know the name of right now is this guy right here called the superior sagittal sinus. You'll learn the rest of them later. So that's the periosteal layer and the meningeal layer. Between the 2 are those dural venous sinuses. Now the meningeal layer is what you are seeing right here. So if I took a brain out of a skull, and I have many times, the periosteal layer stays with the skull and the meningeal layer comes out of the brain. Does that make sense? It's an easy way to tell the difference. So if you're looking at this right here, which layer are you looking at? That's the meningeal layer of the Dura Mater. Does that make sense? Okay. So this is the meningeal layer of the Dura Mater. It will come out with the skull. However, the meningeal layer of the Dura Mater is actually sort of attached to the crista galli of the ethmoid bone. So if I want to get it out, I actually have to cut it off the crista galli of the ethmoid bone. Does that make sense? Okay, so let's talk about these. So the inner layer of the Dura Mater is called the meningeal layer. It forms the external covering of the brain. And then it extends into the brains in the biggest folds. So you guys know, you've had a brain for a while, you've seen brains, you know that there are some big folds in the brain. And one of them is this big, huge fold right here that goes between the two halves of the cerebrum, right? You guys know that that one exists, right? Okay, so let me show you a couple of them. So, there we go. Okay. You might not have learned the name of this before. Do you know the name of it, anybody? The big fold between the halves of the cerebrum? I would guess that too, but it's called the longitudinal fissure. Okay, so here's what I want to tell you. That opaque sac, that meningeal layer of the Dura Mater, it's not just a sac. It also has partitions that go into the biggest folds of the brain. Does that make sense? Okay, so it's got a big fold that goes in through here, and it goes into that fold, right? It's got another big fold that goes between the two halves of the cerebellum, and then a big fold that goes between the cerebrum and the cerebellum, okay? Those are called cranial dural septa, okay? Let's break that down for a second. Cranial for obvious reasons. Dural because what are they made of? Dura Mater, right? And what does septum mean? Partition, yeah? So these are cranial dural septa. So let's look at them, okay? So what you are seeing right here, the stuff that isn't blood vessels, are the cranial dural septa. These are super hard to picture until you actually have a 3D head with you. So next week when you start on the brain, on each campus, make sure that you don't just look at the brain that's in front of you, whether it's human or sheep brain. Go to the cadavers, because we've done on each campus, there's at least one cadaver that has the cranium that you can remove, and then you can take out half of the brain. When you take out half of the brain, you can see all of these. It's really hard to picture until you actually get that 3D image. So make sure that you get out the cadavers next week, okay? If they don't remember, ask nicely and they'll do it for you. Okay, so there are 3 that you're gonna have to learn, actually 4, but nobody ever tests over the other one. This guy right here goes between the two halves of the cerebrum, and it's called the falx cerebri. Right here, same one, right? It's very easy to see. If you took out one half of the cerebrum, you'd see this big partition, right? This little guy right here, do you see that little one right there? Okay, that little one right there goes between the two halves of the cerebellum. That's called the falx cerebelli, okay? And then the one that is really hard for me to picture because I can't figure out what the hell I'm looking at in this picture because I want it to be 3D and it's 2D, is this one that's going lateral. Do you see that one back there? It's going this direction. That goes between the cerebrum and the cerebellum, and that's called the tentorium cerebelli. And I would not be able to test you on that one for nothing unless I had a 3D image, right? So you're going to be tested on that one in lab, not in lecture, okay? So we've got those 3 partitions, and then there's one more that you won't be able to see unless you're in lab. So it's called the diaphragm cellae. It forms a roof over the sella turcica of the what bone? Sella turcica of the what bone? Of the sphenoid bone, yeah. So those are all of the cranial dural septa, and you're going to need to look at them in lab for sure. The falx cerebri, this guy right here, that's the one that's attached to the crista galli of the ethmoid bone, right? So it's sticking up there. And so if I want to take a brain out of the cranial cavity, I can get it all loose except for that little part, and then I have to pull it up and slice it off the crista galli of the ethmoid bone, or else it won't come out at all. Does that make sense? Okay, so these are called the cranial dural septa. And we do have on each campus both a cadaver, and then on this campus we have a half head, just a head that we did a sagittal section of in a jar. So, which is freaking cool. Don't forget to look at all the human brains next week. A lot of people are just like sheep brains and models. There's 15 human brains on each campus for you guys to look at, which is awesome, right?
These are the folds, right?
What?
Those were the folds?
Folds, yes, exactly. They're the folds of Dura Mater into the biggest portions of the brain. Okay, now let's add a little bit of spaces. Okay, so we had an epidural space and we've got the Dura Mater. And then this space right here just has a little fluid in it, and it doesn't really matter, but it does have a name. It's called subdural space. And then deep to the subdural space, we're going to run into the arachnoid, yeah? Okay, let's look at the arachnoid more closely so we can figure out what we're actually seeing there. So if I look at the arachnoid, so watch here. So orient yourself again. I had the periosteal layer of the Dura Mater and then that blood vessel, right? And then the meningeal layer of the Dura Mater, right? That makes sense? And then the gray below that is going to be the arachnoid. Okay, so I'm going to blow that up. This picture right here blown up is this picture. So orient yourself again. Periosteal layer, blood vessel. Meningeal layer, right? And then this is going to be the arachnoid. Remember how I told you it was spider webbing? Do you see how it's spider webbing? Okay. What the arachnoid really does is it has a whole bunch of collagen fibers that are attached to the Pia Mater, and then they're attached to the bottom of the Dura Mater, the meningeal layer of the Dura Mater, right? So that's why it's called arachnoid, because it's like spider webbing. Now, this space within the arachnoid, they call the subarachnoid space. But it's really the intra-arachnoid space, is it not? So it's got a whole bunch of spiderwebby fibers, so I'm telling you that this is the arachnoid, right? And the space within it, they call the subarachnoid space, but I protest that. I say it's the intra-arachnoid space. Why the hell do you care? Because cerebrospinal fluid circulates in the subarachnoid space, and it's really freaking important. We'll talk about cerebrospinal fluid. Does that make sense to you guys? So this is the subarachnoid space. It's the site for cerebrospinal fluid circulation around the outside of the brain and the spinal cord. You have a subarachnoid space that goes all the way around your brain and then all the way around your spinal cord, and it circulates. Okay. Also, there's a whole bunch of room in here, so there are a bunch of big blood vessels that go through the subarachnoid space as well. So I'll show you the same figure again. Okay, so this picture that you're looking at, the Pia Mater right here has little bitty blood vessels underneath it, right? So that's going to be the Pia Mater. These big guys right here were probably running through the subarachnoid space before they pulled it off. Does that make sense? Okay. So the subarachnoid space has bigger blood vessels in it than the Pia does. Okay. So the arachnoid is going to be really important in the subarachnoid space because this is going to be the site of cerebrospinal fluid circulation, and it's got these little projections. Do you see this little projection that goes from the subarachnoid space up into this blood vessel? Individually, that's called an arachnoid villus. Here's what it is. It's a one-way valve. When the pressure starts to build up in the subarachnoid space, it will push that valve open, and you will be able to dump excess cerebrospinal fluid into that blood vessel. So now the blood vessels that we learned, those dural venous sinuses, are not just draining blood from the brain, they're also draining cerebrospinal fluid from the brain, okay? And it'll all go into the jugular veins. Is that okay? And we're going to do that circulation in just a second. We're going to do the cerebrospinal fluid circulation. So that's called an arachnoid villus. Hold it in your head, and we're going to come back to it in a second. Okay, so we've got our subarachnoid space. And then the Pia Mater we already did. We talked about the Pia Mater, which is tightly bound to the surface. So you really can't get the Pia Mater off the surface of the brain without damaging the brain tissue. So if I start to pull the Pia Mater off the surface of the brain tissue will often come with it, right? So for all practical purposes, the Pia Mater is just the shiny surface on the brain. So it's kind of like a serous membrane of the brain, although it's not really very wiggly. Okay, so now one more time, let's just go through the spaces and meninges. So around the spinal cord, but not the brain, we have an epidural space. What's in the epidural space? Fat, yeah, fat and loose connective tissue, yeah? And then we run into the most superficial of the meninges, which is the what? Dura Mater. If you're around the brain, the Dura Mater has two layers. What's the most superficial layer of the Dura Mater around the brain? Periosteal layer, yeah? And then the deeper layer around the brain is the meningeal layer. Okay, don't have two layers down here, it's just one Dura Mater. Deep to the Dura Mater is a little layer of really just interstitial fluid called the what? Subdural space, yeah? And then deep to the subdural space, you run into your next, the next of the meninges, which is what? The arachnoid. The space within the arachnoid, but is not called that, is called the what space? Subarachnoid space. Does that make sense? And then deep to the arachnoid and the subarachnoid space is the meninges that is stuck to the surface of the brain. This one, what's it called? Pia Mater. Got it. Good? Okay. So those are our 3 layers, in addition to bone, that are protections around the brain. Now, the next protection around the brain is cerebrospinal fluid. So your brain is floating in fluid. It's also filled with fluid on the inside. And that floating in fluid helps to protect it both chemically and physically. Physically, it's protected because if I get hit on the side of the head, it is actually sitting in fluid, and so it's kind of cushioning it from jolts. So do you get a concussion every time you get hit in the head? No, you don't, because you've got fluid around it, right? You have to get hit pretty damn hard in order for it to smack against the side of your skull, because it's basically floating in fluid, and so is your spinal cord. So that's its physical protection. And then in addition, the cerebrospinal fluid is also chemically protected, because what the cerebrospinal fluid is basically the richest juice possible. Lots of glucose, lots of vitamins, lots of the proper ions, lots of oxygen in it, right? And it's bathing your brain from the outside and also from the inside. So it's both physically and chemically protected. Does that make sense? Okay, so now let's talk about how you make it and where it's circulating. Okay, first off, you may or may not be aware that your brain is hollow. Your brain and your spinal cord are both hollow. During development, unless you have spina bifida, what would happen is, well, it starts out with this way for everybody, even people who have spina bifida. Your nervous tissue, your embryonic nervous tissue starts out flat. And then as you mature, what happens is it curves and it closes, but it leaves a hollow space in the middle, right? And that hollow space inside the brain looks like that chicken head. See my little chicken head, right? It's a chicken head with a bouffant, right? So inside the brain, you have these spaces called ventricles that are continuous with a space inside the spinal cord called the central canal of the spinal cord. Is that okay? Okay. So that has cerebrospinal fluid in it, and so does the subarachnoid space. So what we're going to learn now is what cerebrospinal fluid is made of, where it comes from, how you get it from inside the brain to outside the brain, and how you get rid of it. Okay? All right, so the spaces inside the brain are called the ventricles. Let's learn their names because it's really easy. The 2 that are on the sides are called the lateral ventricles. You're seeing them right here. You can see them right here too. Can you see why they would be called lateral ventricles? Because they're lateral, right? Okay, you've got 1 in each of your cerebral hemispheres. They are also called 1 and 2, but nobody can ever remember which one's 1 and which one's 2. So lateral ventricles is fine. But after you've learned 1 and 2, you can understand why the next one that you run into is going to be 3. So here's number 3, the chicken head. Yeah? That's number 3. And then I have this aqueduct that leads from 3 to 4. And depending on your preference, you can either call it the cerebral aqueduct, way easier to spell, or the mesencephalic aqueduct. Mesencephalic is more correct, because it's actually running through the mesencephalon and not the cerebrum. But I'll let you get away with cerebral. It's fine. Either way, if it's the aqueduct, that's what you're talking about, okay? And then you come into the last ventricle, and the last ventricle is between the cerebellum and the brainstem, that one right there. And that's called the 4th ventricle. So 1, 2, or 1, 2, I don't know. 3 in the middle, and then 4 down here, yeah? And then 4 is continuous with the central canal of the spinal cord. Is that okay? Okay. So now let's talk about how you make cerebrospinal fluid inside those ventricles, because it's actually all made in the ventricles. It just doesn't all stay in the ventricles. So the brain ventricles are lined by cells that we learned about before. And let me remind you what those are. Huh? Glial cells, but there's a specific type of glial cell that we looked at before. Do you remember the glial cell that I told you guys about that is responsible for lining the ventricles and making cerebrospinal fluid? What are they called? They're called Ependymal cells, these guys right here. right? Okay, so here's what's going to happen. So do you see that those ependymal cells are now lining that hollow space, right, that ventricle of the brain? What the ependymal cells do is they're usually backed right up against capillaries, okay? And what they do is they pull all of the best stuff from the blood plasma. Tons of glucose, tons of oxygen, tons of the ions that you need, okay? And they basically dump it into the ventricles of the brain. So it's basically like the super nutritious juice that you dump in there, okay? It's pretty much just like blood plasma extra, right? And they dump it inside as cerebrospinal fluid. You only ever have about a half cup of cerebrospinal fluid at any given time, both inside and outside your brain and your spinal cord. If it varies much, if you get an extra tablespoon, you're gonna have the worst damn headache of your life because there is no room for swelling in there. If you lose a tablespoon, like for instance, if you have to do a spinal tap, you're gonna get the worst damn headache of your life because you screwed up the pressure inside your brain and your spinal cord. So it's really tightly controlled and it's continually made. Now this is really important because in your body, generally when you're making solutions or fluids, if you have enough of it, there's a mechanism to stop, right? So I don't just continue making blood when I already have, 5 liters of blood. But there is no mechanism to cut off cerebrospinal fluid circulation or cut off cerebrospinal fluid production. That means that the circulation is pretty damn important, right? If you can't stop making it, then getting rid of it is just as important as making it in the first place, right? So what we're going to do now is we're going to figure out how it's made and then how you get it circulating and what happens if you didn't. Okay, so the ependymal cells. So let's look here. All right, so I hate this picture, but it's the best one that they have. Okay, so in the walls of the ventricles, if you look at the models in lab when you get to it, and I'm going to bring models for you guys to play with when we're starting to talk about brain next time. Do you see this little thing on the side of this ventricle that looks kind of like a booger? See a little booger? Okay. What that is a lumpy collection of ependymal cells plus the capillaries they're stuck to. And that's the thing that actually makes cerebrospinal fluid, and you have them in all 4 ventricles, 1, 2, 3, and 4. Got a little booger thing on the wall. And that is called a choroid plexus. So let me show you a choroid plexus in a real brain real quick. I don't know why the hell I closed down all of my stuff. Just, where are you? Chapter 15. There we go. Okay, so I'll show you a choroid plexus in a real brain real quick. There we go. Okay. So, do you see this little booger thing on the wall right there? That's a choroid plexus. Choroid plexus is ependymal cells plus capillaries. Function of choroid plexus makes cerebrospinal fluid. So what it's going to do is pull stuff from the blood plasma and then it's going to dump it into these ventricles. These are the lateral ventricles, right? Okay. So this right here is showing you a choroid plexus, capillaries plus ependymal cells. Yeah? Okay, so you are going to make cerebrospinal fluid in all 4 of your ventricles, okay? That's the only place that you make it. And so what we are going to have to do is we're going to have to circulate it. So let's do the circulation. First we'll do it visual and then we'll do it in a flow chart. So that regardless of what your specific preference for learning is, you'll be able to do it both ways. Okay, so we're going to start with this picture, okay? And we're going to acknowledge that you can't see all the ventricles in this picture because it's a mid-sagittal section. So which ventricles can you not see in this picture? Yeah, you can't see the lateral ventricles in this picture. And the lateral ventricles are the first place that cerebrospinal fluid is going to be made. So you've got lateral ventricles. So we can see them in this picture if you want to, right? Lateral ventricles 1 and 2. On the wall of a lateral ventricle is going to be a choroid plexus that will be making cerebrospinal fluid. In order for the cerebrospinal fluid to get out of the lateral ventricles, it has to go to the chicken head, which is the third ventricle. Yeah? So all of the stuff from the lateral ventricles, the only way for it to get out of the brain is to go to the third ventricle. Now we can see the third ventricle in this picture. The third ventricle is right here in this picture. Now, so why does it look like this? So this picture is showing you physical structures, but it's trying to demonstrate to you spaces. So what you're really seeing is the ventricle is really the one half of the brain is concave and the other half of the brain is concave. So when you put them together, you get space between them. And that's what you're seeing there in the middle, right? That space. So this third ventricle right there. So all of the stuff from 1, 2, and 3, the only way for it to get out of the brain is for it to go through the aqueduct, right? So I made some in 1, 2, and 3, and now it has to go through the aqueduct. And then I'm going to make some more in 4. That make sense? Okay. So now I've got cerebrospinal fluid from 1, 2, 3, and 4. What happens to it? There's 2 possible fates. Stay in the central canal of the spinal cord, which is continuous with 4, right? Or this is how I get into the subarachnoid space, which is the other place that cerebrospinal fluid goes. And what I do is I leave these little holes at the base of my brain that lead from the inside of the brain to the subarachnoid space. And they're called apertures. Yeah? Lateral and median apertures. The median one's the only one you can see here because you're on a mid-sagittal. But let's look at this picture. So median aperture right here and the lateral apertures go out this way, right? Those are going to lead into the subarachnoid space. Okay? Now, let's go back to this figure. So now do you see I'm going to lead into that gray subarachnoid space? Okay. Now, the cerebrospinal fluid is going to circulate in the subarachnoid space all the way around the brain and all the way around the spinal cord. Okay? But I never get to stop making it. So I damn sure better be able to get rid of it when it starts to build up. So the way that you get rid of it is, again, through these little critters that I introduced you to before. Okay? Those are called arachnoid villi, singular is villus. And one more time, what's going to happen as the pressure builds up, as the fluid starts to accumulate in the subarachnoid space? What's going to happen? It's going to push the valve open, and you are going to dump the excess cerebrospinal fluid into the bloodstream. That make sense? Okay. So, what happens if you can't circulate it? What happens if it gets stuck inside the brain? Any idea? It depends on when it occurred. If it occurs during fetal development, you will get hydrocephalus. Okay? So what happened with this baby is the cerebrospinal fluid was continuing to be produced, and it got trapped in the brain. And of course, during embryonic and fetal development, the skull isn't fully formed yet, right? So if you put pressure on the skull from the inside, right, it's going to increase the size of the skull. That's the least of that baby's problems, because in addition to increasing the size of the skull, you are also putting pressure on a developing cerebral cortex, and it is going to cause brain damage, like almost invariably going to cause brain damage. So you're increasing the pressure with this baby during development. So this is called hydrocephalus. Those of us who have been moms know that part of the reason that they do all that measuring every time you go in for a sonogram, they measure the size of the baby's head, but not just the size of the baby's head, because you can have some big-headed babies, right? They'll measure the size of the baby's head with reference to its femur, right? To see if the head is just big, because you got a big-headed baby, and I had a big-headed baby. Oh my God, his head was totally square. It was the cutest thing ever. I should show you a picture. Oh my God. Does anybody else have a square-headed baby? Oh my God, they're so cute. It's like a little block. And I didn't deliver him vaginally, so it was fine. So it was a C-section. So they measure to see whether the size of the head is increasing. So a lot of times what happens with people now is they say, okay, so the head is too big. Do they go in and do some sort of intervention when they detect hydrocephalus? FYI, it's more likely to cause a miscarriage. And also, there's not a damn thing you can do about it. By the time you diagnose the hydrocephalus, the damage to the nervous system is already done, right? So all of these valiant measures that you wanted to go through wouldn't change the outcome almost at all. So this is called hydrocephalus. And I want to show you just this picture of hydrocephalus. So this picture is showing you that the fluid actually builds up on the inside. So this is the size, hold on just a second. This is the size that your ventricles should be relative to the size of the cerebrum, okay? This is a post-mortem photo of a baby's brain with hydrocephalus. Do you see the size of those ventricles? Yeah. if hydrocephalus or if a blockage occurs, where do you think in that whole process it's most likely to occur? What? Where is the one place that's pretty skinny to begin with? The aqueduct, yeah, the aqueduct, right? So if it occurs in a baby in an aqueduct, it gets stuck between 1, 2, and 3, right? If it occurs in an adult, like if I, for instance, had some kind of brain damage or an infection that caused scarring to close up the aqueduct, they'll just stick a shunt in there. They stick it through one of the apertures, up the aqueduct, and then they drain the excess fluid either into your subclavian vein or into your peritoneal cavity. And then as you grow, you just get a longer shunt. Okay? All right, I will see you guys on Wednesday. How long does a baby live with hydrocephalus? Sometimes they can live into adulthood, but usually they'll die in infancy. Yeah, it depends on how severe the damage was. You can have just a little bit of hydrocephalus and a lot of times they'll completely recover from that. But if it's massive amounts of pressure like that, sometimes the damage is so severe.